I come from a mathematics background. I started graduate school in math, but then I realized that I love the applied world too much. And I got my PhD at Stanford in statistics. I had a brief career as a statistician and then again took the plunge back into biology. Actually worked in the biochemistry department for a few years. And then in the past few years have stepped back to really become just a almost pure data science statistical research group. I have a variety of interests beyond math, stats, and biology, including history, as you will see a little bit. But I think that interest in history and history of science has led and influences a lot of my work. Another anecdote is I actually don't know how to use a QR code, so I hope this one is accurate, but I'm just trusting it because someone gave it to me. Okay, so I like to think a little bit about and encourage us to think about what genomics does today. And I'm going to claim, possibly controversially, that the workflows that we are accustomed to and that are bread and butter actually distort a very rich signal that we can make more of, okay? So today genomics is, of course, foundational to discovering biomedicine. I would argue that we are still operating in a paradigm of analysis that was designed for a different era of data. So it was not designed for the ultra-high throughput, precise resolution genomic measurements that today are ubiquitous in biology. Okay, it is not fully statistical or mathematical as a discipline. And the way the field of genomics is organized does not provide a unification across micro niches of biology and their applications. And part of the reason for this is that we are still focused on two pillars of genomics that are focused on references. And I would say those two pillars are assembly and alignment, and I will talk a little bit about both of them today. For this reason, we are missing biology. We are missing the opportunity to discover new biology. And in the papers that I don't have time to go into, I don't have and won't go into these details, but we have empirical proof of this. Many times, the workflows that I mentioned, assembly and alignment, these pillars, they're also computationally intensive and possibly unnecessarily. And then finally, the workflow of genomics today does not provide our field, in my view, with truly AI-ready data. And I hope I can convince you of that at the end. So I think as a field, we are interested in using genomics to access. It's not just, well, it is interesting technical questions, but really what are we after? We're after biological knowledge. Okay, so what is this a picture of? Any guesses? What? Yes, it is entrance to a pyramid, okay? The entrances to pyramids, of course, were obscure. And I think there's an interesting analogy. Okay, so today we think the entrance to the pyramid of the knowledge, that the jewels in the temple, are accessed by assembly and alignment. And then within, and you know, those are the major ones, and then multiple sequence alignment is another way to access some of this knowledge. What is this knowledge? Well, some of it is, we know today, actually actionable biomedical knowledge. For example, in human genomics, we have splicing, mutations, understanding antibody repertoires, and more. In the microbial universe, we have microbiome, and we have bioengineering tools that have been discovered, like CRISPR and other things. Okay, so we also can use multiple sequence alignment to discover evolutionary principles in phylogeny. And then we are starting to see some of the treasures in the temple with deep learning, such as protein structure prediction and large language models. But I think we acknowledge, and Jan gave a great talk yesterday, that the limitations of what we can do today with the way that we treat genomics data in deep learning. So I again think this is an interesting analogy that we're trying to access what's in the temple. We actually may not know. And even today, people are still discovering in ancient Egypt new chambers in very well-studied temples. And I think in biology, there's a huge amount to discover that we just aren't. Okay, so let's step back and think about why that might be. So one of the principles of genomics today is alignment. Raw reads, rich raw data, it's actually a statistical sampling of the true underlying biology. Reads are the first step in the way we analyze them is to use a computational procedure, an algorithm, to match them to the genome. And that is an algorithm that today, that algorithm is not analyzable in the sense of having provable properties. Okay, so that results in several scientific flaws or drawbacks. One is that absent sequences in the assembly, of course, cannot be interpreted when you align. They're simply not there. Raw data is reduced to positions in the reference. And there's also an introduction of biases because there is no provable guarantee that an aligner takes a raw read and has positioned it correctly. Okay, in a formal statistical sense, after alignment, the resulting statistics, even if they're correct, the positions are correct, they are not sufficient because of the problem I just described. Other tasks that we today want from alignment, such as gapped alignment, discovery of de novo mutations in indels, or RNA editing events, they have even more problems and less tractable less tractable framework. So alignment, in addition to that scenario where I didn't even bring up the idea that the reference could be wrong, in human diseases, and especially cancer is a great example of this, the human reference genome is a very poor approximation of the real genome. So alignment to a reference genome in the context of cancer is really inaccurate because the cancers, one of their hallmarks is an incredibly diverse, rearranged genome. Tumors literally shatter, and recombine their chromosomes. Okay, so a reference genome does not exist for even a single tumor. Even if you had a perfect approach to reconstruct and assemble a tumor genome, there isn't a single tumor genome, even for a single individual patient. So references simply, that paradigm of assemble and align cannot capture the biology of tumors. And that idea extends to other diseases. Even a single individual does not have a single personal genome. Apart from human and human genomics, the extent of diversity outside of the most well-studied organism, us, is so vast that it is impossible to imagine being able to catalog every reference genome and store it, let alone to align it. It's simply too much computational burden. Okay, so after assembly and alignment today, the next step is this diaspora of custom bioinformatics that are designed for important but very field-specific tasks, such as defining splicing that occurs perhaps at single cell level, or discovering structural variants, mutations, and so on. And this has resulted in highly specialized labs, algorithms that don't have analyzable properties, and ones that cannot generalize. It's incredibly time-consuming from a computational perspective and also a human perspective, and again, does not have formal statistical guarantees or control. In addition to the lack of a unified featurization for deep learning. Okay, so what I'm going to tell you about for the rest of the talk is a new approach to genomics that is now published. It's called SPLASH. How is it designed? And it's going to be now like a thought experiment along with leading us through why splash is a provable algorithm in the sense that, or analyzable algorithm. Okay, so let's think about some of the zoo of tasks, and I love zoos, zoo of tasks that custom bioinformatics attempts to approach. Okay, splicing, mutation calling, structural variants, and whatever your interest is. What is a conceptual feature of this, of each of those tasks that perhaps has there is a unified formulation of how to detect each of them, okay? So these are some that you may be interested in. Okay, so the claim is that each of the tasks I mentioned, and if you don't have a favorite, think about trying to detect mutations that are, personal genetic variants, for example. Okay, so the claim is that a personal genetic variant, or one in a virus if you're interested in viruses, there is something kind of fundamental that doesn't have to do with reference genomes or the species. So that is, the claim is that what is a mutation? Okay, on one hand, it's a DNA, it's a DNA base pair change. On the other, what it is it's a difference between people that can be measured in the language of ACGT, okay? And somehow it's a regulated change because some people, and if it's a disease-causing change, it will be, or an important change for adaptation, it will be selected for. So it's not a random change, okay? So if we're interested in finding such variants, we should be able to use a statistical test to find them. And that change should ideally be discoverable with almost any technology. And it should, this idea of like the signal we're trying to detect should allow us to discover both known events like mutations that I described, but also ones that may not exist. Okay, so let's think about if we could detect such features directly by understanding raw sequence reads. My claim is yes, and I'm going to walk you through what splash is. So splash is flipping the paradigm of what genomics does today from interpret first and then do statistics to first do statistics and then interpret. And when I say first do interpretation today, what I mean is reference-based. So you assemble and align. That is an interpretive step. So what exactly is splash? It essentially catalogs every k-mer in every FASTQ file that is provided to it, and it looks for statistical, and the k-mers here are going to be colored, and it looks for statistical patterns or deviation from the null model of the next k-mer is drawn IID from a multinomial. I will describe that in a little bit. So at a high level, you can start to think about splash is going to be an algorithm that looks through, exhaustively catalogs all k-mers in all raw sequencing data, performs a new statistical test of IID-ness of the k-mers, and then identifies sequences that are likely to be regulated. This work was published last year in Cell, and then it also has other papers that are both on the bioRxiv and in other journals. So let's go through a very specific example. Suppose there are two strains of a virus. We don't know anything about the virus, okay? So we have Delta or Omicron. We're going to make a definition, a mathematical definition of strain. Interestingly, when I talk to virologists or microbiologists, they generally don't have a formal definition, like a mathematical definition of many of their terms, including strain. But let's just define it to be two viruses or strains of each other if they share more than 99% of their DNA. So if there are two strains of a virus, what gives us the existence of two strains is that there's a large amount of similarity in their sequences, and then there are these critical key differences. So in this case, a SNP. It needn't be a SNP. Okay, so what we're going to say is if there are two strains, let's say there's Delta and Omicron, then there exists some sequence in blue, we'll call this an anchor sequence, such that downstream of the anchor sequence, there exists another sequence, we'll call this the target sequence, that differs. Okay, it's a pretty simple idea. Okay, so this idea hints at a mathematical algorithm that can detect strains without knowing anything about what the sequence is. Because we're just saying if the two strains exist, then there is some sequence, there is some blue sequence. We don't need to know a reference for it. Okay, so that's essentially what SPLASH is. We don't know what this blue sequence is. If there are two strains, we just know it exists somewhere. Okay, so we exhaustively look across every single possible sequence that could be the anchor. Then we catalog the targets. In the following toy example, we suppose that there are 4 individuals infected with different strains at different levels. Okay, so B only has strain number 2, and so does C. D has mostly strain number 1. And so we build a contingency table of counts, which are, for a fixed anchor, we have 1 contingency table. That contingency table, in this case, has 2 rows, the 2 different sequences after the anchor. And then it is populated by the number of counts for each k-mer observed in the raw sequence data. After this, from the biological interpretation is, okay, are the people's counts IID? The vector of counts, are they IID draws from a multinomial distribution? Does anyone have a guess at what test we would do to try to test that hypothesis? Chi-squared. Okay, any others?
Yeah, the counts are too small.
What's that?
The counts are probably a bit small, I guess.
Chi-squared is a great idea because, okay, there could be tons of different, I've actually simplified this. There are going to be a lot of targets. Many times there are lots of counts. There are lots of samples. So I actually spent a fair amount of my PhD studying chi-squared and its technical properties. And it's one of the most widely used test statistics out there. However, it has some important problems that are shared with many other statistical methods from that era of data and data analysis. Does anyone know one of their properties that's negative for this application? Okay. I am a huge Pearson chi-squared nerd, so I know a lot about this. It's incredibly sensitive to subtle deviations from the null. Okay, so what it is testing is each column is drawn from a probability vector, P, and it's testing whether PI, and it's testing the equality of the PIs. If one PI has this epsilon difference from the others, it will readily reject, especially with small counts. From a biological perspective, we don't want that property because, first of all, it's uninteresting if there's just a subtle deviation of one of these column counts, and second of all, technical problems can really easily distort that strong null. Okay, so we developed a new statistical test that's actually much more generally applicable than to SPLASH for this purpose, and that is a workhorse here. And we've actually shown that if we use Pearson's chi-squared instead of the OASIS test, which is what it's called, the signals that we find are they're not very interesting, and they are driven by technical noise, whereas SPLASH in our hands is not, and provably so. Okay, so this whole framework and idea leads to a proof almost, or probably a proof, that SPLASH finds mutations, and not just mutations, could be indels, any variation in sequence defining strains with no reference genome under conditions that can be completely spelled out, okay? So I mentioned this. The first steps in SPLASH are to take raw sequence data, FASTQs. It doesn't need to be FASTQs. They are then processed so that each k-mer in each read, in each file, is chosen as an anchor. For each such k-mer, we build a matrix of sample, could be a person, but it needn't be, by target, could be one base, could be 27 long, you choose. And then we perform this, we build the contingency table and perform the statistical test, okay? So here's a quick kind of motivation for why this is so general. I presented the idea of an anchor being able to detect mutations in a virus and strains. But you could replace this target sequence by an exon, not just a mutation. Okay, so suppose now we have a 3-exon gene. Exon 1 is in blue. Orange and purple are exon 2 and 3. Then the target sequence isn't a mutation, it's an exon, and we are looking for variation in the relative expression of exon 1, 2 and exon 1, 3. Okay. So if you kind of step back and ask, what are the critical ingredients for this test and framework to work? They're very simple. There's just one of them is that there's more than one variant. And the second is that the variants are regulated. They're not drawn IID across each sample. And these two ingredients are very, very simple, and they're also general in the sense that they apply to a vast number of different biochemical mechanisms in genomes, including alternative splicing, mutations, discovery of VDJ recombination, so antibody repertoires that are the reason that you can be vaccinated. Your adaptive immune system can be trained to recognize a pathogen and then protect you. All of these features can be detected on purely statistical basis with absolutely no knowledge a priori of any mechanism causing any of them, let alone what the sequence is. Okay, so again, SPLASH is a very general, unified statistical detection of sample-specific sequence variation. Sample is arbitrary. In spatial transcriptomics, sample is a spot on, it's a barcode. It's a spatial location in a tissue section. In single-cell 10x, a sample is a barcode. It could be a time point in a longitudinal sample and more. Sequence is also very general. We currently have it implemented for nucleotides, so DNA, RNA, but it is more general than that, actually. And so together, this provides a very kind of streamlined workflow. You take raw, and it's a single line command. It is super fast and super easy to use. So you just provide a list of your raw FASTQ files. You write splash space the file name, thanks to fantastic collaboration with Sebastian Deorowicz's group, including Marek Kokot and others. The algorithm is ultra efficient now. It identifies the anchors that are have this evidence of statistical variation across samples. And then it opens the door for a variety of interpretation steps that I think, to me, are among the most exciting today. Okay, so in my research. So these k-mers that I described to you, anchors and targets, are very short. They're 27, but together they're 54. actually, there's a huge amount of mileage that you can get from 50-formers. And that was that was reasonably long in the early days of Illumina sequencing. But arguably, they aren't long enough for full interpretation. And so what I want to talk a little bit about now is what we've done, what we can do after Splash to interpret and find potentially evidence of function. So one of the things that we have done is to develop an ultra-efficient seed-based assembly that extends the anchor target to a much longer context. Okay, I'll talk a little bit about that. With or without this step, we have a variety of options available today for post facto interpretation of the anchor targets that either use alignment or completely bypass it. So in the simplest framework, everyone could use Splash as a pre-processing step, just like you use read trimmer. You could run Splash and replace your raw data with Splash anchor targets. The advantages of that, there are two major advantages. One is speed and data size reduction. So that step is intrinsically a highly compressive step. You have essentially removed all sequences in your data that have any evidence of, or have no statistical evidence of variation. So any background boring sequences are gone. And it, in practice, reduces the data size orders of magnitude. In addition, you also are now working with a post-statistically analyzed set of data. You don't have to do any custom bioinformatics statistical tests for differential expression of splicing or differential mutations, because every sequence in your file after Splash there is evidence of that. Okay, so these are some of the optional post facto analyses, but any single post facto analysis that you might do on raw reads can also be done at this step. Okay, I mentioned a reason to do it is it's super efficient. That should be appearing in Nature Biotechnology very soon. Okay, so I want to now talk a little bit about compactors, and they are a different approach to a different way of approaching the idea of assembly. They are not, I would say, importantly, their purpose is, I would say, in the direction of assembly, though it isn't exactly in the direction. Compactors are not trying to replace assemblers and replace the length of the assembly that current assemblers provide, but instead like to just step back and say, why do we do assembly? So there are some complaints and downsides, okay? So assemblers, they can have errors, of course, because they're algorithms that don't have, again, provable properties of when they, with a handful of small exceptions, there have been no proofs of when and under what conditions assemblers misassemble. And so that causes errors and missingness. In addition, the production of an assembly, while interesting, is itself not the production of a biological inference. So it's for example, even if the assembly is correct, you have an assembly, but you don't know the functional elements within it, okay? So it provides an entree into studying many important problems. I'll give you an example of one or two, such as covariation. It provides an entree into studying evolution and evolutionary constraints and pressures on sequence. But in and of itself, it doesn't do that. Okay, so one of the uses of assemblies is as a prelude to multiple sequence alignment, where you can look at how bases in two assemblies are kind of co-regulated or co-varying, and use that idea to infer that they may be, quote, talking to each other in a functional way. And the reason that I'm asking these questions is just sort of to say, well, assembly is one way of getting at them, but maybe there's a more direct access route to this treasure. Assembly is also, I mean, it's a necessary step today for multiple sequence alignment. Okay, multiple sequence alignment, in addition to having this, like direct entry, potential entry into function, I mentioned with covariation, it's critical to some of the deep learning applications that we want to use it for today. For example, protein folding. The MSA in the ablation analysis of AlphaFold was the most critical piece. And I would argue that piece is highly approximate. I'd be happy to and interested to talk to people about that. Okay, so what are we doing instead? And I'm just going to wrap this up quickly. First, I'm going to tell you about a proto-assembler, I'll call it, compactors. So compactors is it's an alternative to assembly at a small scale. It is a statistically guided algorithm that, like Splash, has provable properties for when and how it produces these longer range sequence contexts. And it has a bunch of different parameters and an underlying probabilistic model that, for time reasons, I will not go into. But it makes a statistical decision about when to extend and how. So if the anchor is the dark color and the target is this cross-hashed color, what the compactor does is it says, okay, I'm going to try to go further than the target. I'm going to go another 27. How am I going to do that? Well, in many cases, this context length falls within a read. So that is relatively simple. But how is it going to decide if it will, okay, so one question is, how will it decide to split, okay? Maybe after this target, there's actually 2 different assemblies, or maybe there's 1,000. It performs a statistical test that says, okay, I'm gonna start with the most abundant compactor, the most abundant extension. And then I'm going to test if I added, are the rest of the sequences that I observe compatible with sampling that most abundant sequence with noise? If not, I'm going to add a different compactor, and I'm going to do this iteratively. Okay, so that's the high level of what the intuition of compactors. Of course, there's a lot more math and algorithm there. And that paper is now in review and also on the bioRxiv. So what can we do with them? So one of the things that we can do with compactors is consider replacing some of today's approach to understanding covariation in sequence. So I'd like to compare it quickly to multiple sequence alignment. Today's multiple sequence alignment and specifically for RNA structure, although could be more general than that. So today, how do we infer RNA structure? There's 2 approaches. One is very, very resource intensive, and it's an experimental structure prediction. The other is more computational, and it's based on assembly, then multiple sequence alignment that aligns different variations on the theme of a fixed RNA, such as a ribosomal RNA, actually. It aligns them and then searches for bases that have signatures of change, repeated change, and correlated change. So covariation is literally a correlation in change at different positions. I would say the structure of the ribosomal RNA, which is a critical piece of how the ribosome functions, was discovered or predicted with computational procedures that were an MSA followed by covariation analysis. So they're incredibly important and powerful. But in the step of the pre-processing step to this inferential covariation, we are introducing distortions through assembly, and then further distortions in multiple alignment. So multiple sequence alignment is just more complicated alignment. So what could we do with SPLASH instead, the SPLASH framework? Well, this is kind of an attempt to explain it at a very, again, very high level. 10 minutes. Okay, so I'd like to leave a few minutes for questions. What are we going to do? Okay, the claim is, think about an example of a virus or a bacteria that has a functional element that is rapidly evolving, but that there's a constraint on that evolution because there's a function for some of the elements. Does anyone know an example of a regulatory element like that? There are many in SARS-CoV-2. HIV is, there's the TAR element, there's a frame shift element in HIV. HIV is one of the most highly structured viruses, but that we know of because we've studied it so much. But essentially, every RNA virus has, to my knowledge, has regions that are highly structured and incredibly functional in the sense that if you ablate them, the virus dies, including, of course, influenza, and we could just rattle off the names. Okay, so there's a huge reason to try to detect these structural elements because they're functional, they can be targeted with drugs, and especially in cases of viruses that we've never seen before, we don't even know exist. We would like to be able to find a feature that shows this virus is actually infecting and under selection. Okay, we don't want to just, for example, sequence the wastewater and say, okay, here's a list of all the viruses there. Okay, a public health official would not be able to really, okay, if you compare that information to, we've sequenced the wastewater, and we can tell you there is this covarying, this evolutionary, this sequence that is under selection that you've never seen before. It's a very different scenario. Okay, so that's why we want to detect it. How are we going to do that? Well, we're going to take raw reads, taking the anchor, fixing the anchor as part of a multiple sequence alignment, like kind of the intuition, the same intuition, and then start to examine the target sequences in a framework that would allow us to detect statistically and probabilistically covariation within the targets, bypassing assembly and alignment. Okay, so suppose this is the base target. What we're gonna do in the most kind of first naive approach is simply scan it for evidence of base pairing. That is reverse complementarity. Let's say we find some, this former. Then what we're going to do is we're going to look across the targets and ask, okay, are there any targets that have covarying or mutations at all in their sequence? And if there are mutations, do they exist in the base paired region, or do they exist outside of it? Okay, so in this toy example here, G and C exist in the target in the base paired region, and then C exists outside of it, T exists outside of it, G exists outside of it, and so on. Okay? These numbers can be together used to test the null hypothesis that the mutations in the targets are randomly distributed across this sequence, and they're not trying to preserve this reverse complementarity. Okay? So take my word for that. We could do a whiteboard later. That leads to a direct probabilistic statistical test for selection on structures that bypasses multiple sequence alignment, it bypasses assembly and also alignment. And this is work led by a fantastic graduate student in the lab, Julie Wang, and Arjun Rastagi, who just started his group in UCSF. Okay, here's the math if you want. It should be appearing in the bioarchive any day. It allows us to get a structure score for every anchor based on the targets, and then a better one, more sensitive test based on the compactor. So the same logic could be applied to this longer sequence context. All right, so a snapshot of what we've been able to find is we've been able to re-identify the key structural elements in HIV and other viruses, as well as new viruses that are, or predict viruses, like structural elements in viruses that have not been described before. It's a method that can prioritize elements that are under selection, not just exist. And I think provides a quite exciting, at least to me, general approach to studying evolution of genomes directly from raw sequence data, as well as functional elements. And then kind of just to summarize, other avenues that we are actively working on, and I would love to talk to anyone, and especially looking for postdocs in this area, is using Splash for deep learning applications. After Splash is run, there's a huge opportunity for any kind of interpretation. As I mentioned, some of them, including any current bioinformatics workflow, can be run. There's also a much wider opportunity for application of deep learning. Okay, so check out our website if you're interested. I'd love to talk to people while I'm here or later, and I thank the people that have done this work as well as funding agencies.